All right, so welcome back to Animal Behavior. Today, so reminders here. So if you haven't done it already, that Unit 0 with all of those syllabus type assignments, make sure you complete those, and that should unlock the rest of the course. Gradually, these assignments, I'm building them and they're trickling in. So I think I've got the first three of applied reading exercises done, and the first one of the unit comprehension exercise. So the material for that UCE is covered pretty much half or 75% in this lecture, but then also in the reading. And so you should be able to find a lot of those answers in the reading, but otherwise, from this lecture. And so the rest of it will be on Thursday. So if you open it up and said, oh, I don't know what these things are, well, that's kind of expected because it's meant to be something that helps test your comprehension right at the end of the unit, not going into the unit. So so they'll be due. These generally will be due on a Sunday after the unit is finished. So this upcoming Sunday, relatively short. There's a drop policy. Remember these. So there's going to be one per unit. I think we have 10 units. And so there's a drop 2 policy on the perusal assignments and a drop 5 on the UCEs. So pretty healthy drop policy, but hopefully, it doesn't take too long to complete. And the grace period's through Monday morning. So even though it'll show up on your Canvas as being due Sunday night, as long as you get it in when Canvas allows you to get it in, so until Monday at 10 a.m., then you'll be fine. So any questions about the assignment structure? And aside from the midterm and that final, and then that group project, these are the only assignments. Well, and then this kind of the attendance stuff, so the participation scores, which is why I think I got a drop 5 as well. That's the only, the only points you have to worry about for this course. So there's no additional homeworks or anything like that. Okay, so yeah, cool. And so there's this questions link down here, which you can also get through this QR code. So Anushka, the TA, will be monitoring that. And so if there is a question you'd like to ask, but you don't feel comfortable enough raising your hand, you can post an anonymous question here. And if Anushka sees it, she'll flag me down. Otherwise, I can take care of it after class. So if you click or go to that link, it'll say, hey, do you want to register? You can just click skip, and then you can just post an anonymous question and even vote on other questions. This isn't a big enough class that I think the voting will be important, but but that's an option as well. Okay. All right, so back to where we're going here. So this up here is an octopus, zoomed in kind of on this octopus eye. An octopus is a cephalopod. So cephala, head, pod, foot. So it's basically a big head on a foot. So it's an invertebrate. And cephalopods also come in this flavor over here. This is a cuttlefish. And so I hope you notice in both of them, they have these very striking eyes there. And so those eyes to me look a lot like our eyes. So they're what's known as a camera type eye or a simple eye. It's got kind of a lens out in front, a retina in the back. And, so we look at this, we say the cephalopod eye looks a lot like the vertebrate eye, our eyes. And that's kind of interesting, I think. And so it leads you to want to ask questions about, where did these eyes come from? So evolutionary questions of, do I have the same eye as a cephalopod? Was there a common ancestor that had that eye? And and how could we possibly tell about that? So I can ask a question like this how question here. How did such similar eyes evolve? So that's not quite the most refined causal question, but it's kind of the point I'm trying to make here is it's a question that begins with how or why. And it's asking about causation. It's asking about where did this common eye come from? Or how did the cephalopod get their eye? Or how did I get my eye? Or our vertebrates get their eye? And so, so that's, animal behavior. We always sort of start with interesting observations that lead to causal questions. And so our interesting observation here is the commonality of this eye structure. Apparent commonality leads to this causal question we want to investigate. And a causal question, that's a term we'll see over and over again. question about the cause, usually how or why, and is answered by a statement. So if you ask a question that's answered with yes or no, it's probably not a causal question. It's probably a hypothesis in disguise. So, when I say, how did such similar eyes evolve? I'll show an example of a hypothesis, but, I could say that, cephalopods and vertebrates have a common ancestor with that eye. That would be a statement, that answers that question. Not a yes/no answer, is a statement that I would need to defend. So that's what I mean by causal questions. So before I move on to the answer to causal questions, is there any questions about the causal questions? Is that pretty clear what I mean by a causal question? All right, great. So our goal as scientists is to provide potential answers to causal questions. And there is a fancy name for that, a hypothesis. So a hypothesis is just a plausible answer to a causal question. It's an unconditional statement. How many of you learned that hypotheses are if-then statements? Okay, forget that, because that's wrong. So if-then statements are predictions, which we'll get to. A hypothesis is always an unconditional statement. The if-then is something that follows from a hypothesis in an experiment, as we'll just about talk about here. But a hypothesis is never an if-then statement. Now, hypotheses always have predictions, and predictions are if-then statements, and so it's easy to confuse those. But if you were to ask somebody what's a plausible hypothesis for why the cephalopod eye looks so much like the vertebrate eye, you would not be satisfied by them answering you with an if statement. Like, what does that mean? I asked you, how did such similar eyes? And then if you were to say, well, if, that's just not an appropriate answer to a causal question. So a hypothesis is an answer to a causal question. It is not an if-then statement. So it is an unconditional statement. It's a starting point for an investigation. The word hypothesis is broken up into two parts, hypo and thesis. So a thesis is something you're going to defend. When you write a thesis, then you've written a document where at the top you're going to say, my thesis is this. And what's about to follow is a whole buttload of data and arguments that backs up that thesis. So you are going to be convinced that my thesis is correct. A hypothesis is not quite there. Thus the hypo, not quite a thesis. It is a plausible statement without data. And it is our job to gather data to try to make the hypothesis stronger and stronger. And eventually, if we can defend a hypothesis with data in a wide range of contexts, in a wide range of contexts, maybe over different animals, then eventually that hypothesis hardens and becomes a theory. And a theory is something that we don't need to test. We might test, but we don't need to test. We're then allowed to take it for granted and use that assumption of the I don't want to say truth, but take it for granted to aid us in testing other hypotheses. So a theory is like a hardened hypothesis, hardened with data. But before you have any data, it's a hypothesis, not quite a thesis, a hypothesis, and never an if-then. Any questions about that? Is that a question? No? Okay. All right, so let's come up with a hypothesis to test. Was there a question? I'm sorry. It would be an answer to how. So it provides a how. So the causal question poses the how, and a hypothesis is one of many different answers to that. Does that answer your question? It will respond appropriately to how. So the causal question may actually be how or why. Why is like, why do we have eyes? That's also a causal question. So how or why questions get at causality. And this is like an example. I could say, well, how did this happen? Well, it happened because vertebrates and cephalopods had a common ancestor. I'm not saying this is the case. In fact, I hope you don't believe this because this isn't the case, but I'm just going to assert this right now so that we can then investigate it experimentally and see if we can reject this hypothesis. Ideally, we would have multiple hypotheses. So we'd say, well, it might be that they had a common ancestor. What's another hypothesis that would explain this? Yeah. Can you explain what you mean by convergent evolution? Yeah, so so those who didn't hear it, so the idea is maybe convergent evolution or parallel evolution, where you have similar selective pressures on both cephalopods and vertebrates, and that caused them to come up with a similar solution, but they actually weren't related in the evolutionary history. They just happened to both arrive at the similar solution. That would be another hypothesis about why there's something in common here. It's just kind of coincidence, but it's shaped by the problem they were solving, not by the evolutionary baggage. And so you might say, well, why do I have a similar looking eye as my dog? Well, we're both vertebrates, and there was probably a common vertebrate ancestor that had that eye. But why do I have a similar eye as this cuttlefish? Well, that might be because that eye developed under similar selective pressure as this eye, and it's just kind of a coincidence that they look alike. So that would be another example of a hypothesis that would answer this, and we would hopefully be able to test both of those and be able to sort of come to some conclusion as to which one has more evidence. All right, so any questions about what I mean by hypothesis as a plausible answer to a how or why question? Yeah. So yeah, that's a great question. So a lot of times, the first time you hear the word hypothesis is in a stats class. You talk about the null hypothesis, and for this class, when you put on pause the notion of null hypothesis, a null hypothesis is a technical detail that in the kind of the in sort of the mechanics of looking at data. We don't always have to worry about forming a null when we're answering causal questions. Now that I've said that, in order to figure out where these questions lie, there is sort of a null that, like, if you kind of look out, like in this case, this eye for the cuttlefish here, or for the octopus, or for this human, this child down here, looks so similar that statistically speaking, it seems very odd that this would happen by random chance alone. So that's kind of what I mean by non-trivial. So it's like you're walking out through the desert and and you start noticing that there's some interesting features that seem to be shared between that scorpion and that tarantula, and you start saying that can't just be chance. And so that's kind of the case where it's almost like you use the is it random chance or not to figure out. That's kind of what I mean by non-trivial here. It couldn't be explained by just random chance. And so, what's non-trivial or not is in the eye of the beholder, but so there's a bunch of things that can motivate why you ask a causal question. But this is just kind of my rough guideline of where the causal questions come from. It's from observing nature and noticing, that's an odd pattern. And so that's what I mean by non-trivial. It's a notable pattern. Does that make sense? Any questions about that? All right. And this happens in behavior all the time. So you see an animal doing something and you're like, well, that animal could have just bumped into something randomly, but it keeps doing that over and over. So it seems like it's not just random. What's going on there? Let's pose a causal question to see what the cause of that is. Okay. All right, so we got our hypothesis that we want to test. So, because we're talking about phylogeny, and I just want to get some of these terms out of the way, I'm going to put a pause to the eye stuff for 2 slides and just say that, what we've been talking about so far are so-called phylogenetic hypotheses. So these are hypotheses about things over evolutionary history. So how did these particular phenomenon pop up in evolutionary history, and how are they related across different species? So very long time scale is what we're kind of talking about here. So so how many people are familiar with the term phylogeny? Okay, good. So hopefully you've seen it, maybe 181 or 182, you at least talked about phylogeny a little bit. But just for those who aren't, so this phylogenetic basically has to do with this idea of evolutionary timescales and how species relate to each other. And phylogenetic questions often relate to is there, if we notice two things that are similar, did they evolve independently? Was there a common ancestor? And if there does happen to be suddenly a bunch of individuals with that particular trait, bunch of species with that particular trait all around the same time, are there things in deep history that might suggest why that particular thing happened? And so an example of this, and in the reading for the next unit, you'll if you do the AREs, you'll see this for the sweat bees. There'll be a similar phylogeny here. So this is a type of phylogenetic tree. So a phylogenetic tree is one way to summarize a hypothesis about where things came from, about a phylogeny. This particular type of phylogenetic tree is called a chronogram, which basically means that each one of these vertical lines here, the length of them have to do with time back in history before an event occurred. So the idea here is you have this common ancestor. So these are basically all of the Lepidoptera, those are the butterflies, and these are different types of butterflies. And we've got this outgroup here. And so if we see up here that there is this earliest Lepidopteran species, so that's the earliest butterfly, and it is around between the Triassic and Jurassic period right here, say 175 million years ago. And then if you were to look back through, so how do you get these trees? Well, there's a bunch of different ways you can do it. Molecular methods are very popular now. So that's what you'll see in the paper for the next unit about the sweat bees, is that they use molecular methods to sort of say, we've got a bunch of sweat bees here today. And so if I look at their genes, I can see how they've drifted away from each other. And I can estimate at what point did these species sort of start to diverge from each other. And that is kind of where these branches come from. And so based on the evidence they have in the fossil records, you can do this with characters you see in the fossil record as well, you can reconstruct these plausible phylogenies. And there's basically a computer program that uses fancy statistical methods to spit this thing out. And that's its best guess based on the data that you gave it. It kind of looks like this. And you get all of these kind of extant species down here, and then sort of hypotheses about how they're related in evolutionary history. And what this particular reconstruction shows us is that, so these green dots, those are things in the fossil record. Those are examples of these early butterflies. And then down here, we see that after this violet band, you get a bunch more of those green dots across all these other species. And looking at this, when you correlate that with, well, what goes on in this violet band? Well, that is this angiosperm radiation. Those are flowering plants. And so there's a diversification of the flowering plants that occurs around this time in history. And right after that, you start seeing a diversification of the butterflies. So this phylogenetic hypothesis starts to answer these questions of why do we have such diversity in butterflies? Well, it suggests that it was driven by an initial diversity, a growth in diversity in these flowering plants. So that's an example of, a more complex phylogenetic hypothesis. Yeah. Lepidoptera and butterfly. Right. So, Kendrick, do you happen to know what trichoptera are? No, that's all right. That's fine. So, yeah, the way these are usually drawn, it's usually consider a very closely related insect that's not the insect. So they're saying these are our butterflies, but we want to know, where did the butterflies come from? And it's saying that not that Trichoptera is the ancestral state of a butterfly, but it comes from, so they had a common ancestor that wasn't a butterfly or maybe a Trichoptera, and all these were butterflies. And so they're trying to capture the emergence of butterflies, which is why they include that so-called outgroup. So so that's why there's this extra one here. I also want to make sure that I emphasize this is not a ladder. So I don't want to say that like Trichoptera, because they're connected back here, are like proto butterflies. And as you walk this way down the tree, you get more and more butterfly-like. And this is like a poor butterfly, and this is a good butterfly. These are all extant species doing their own thing, adapted for their own habitat. And there's not like it's we don't it's similar with the great apes. You wouldn't want to say that a chimp is a proto-human or something like that. We can tell a lot about common ancestry and how things came to be, but we don't want to say that just because we share a common ancestor with something that's way back, that thing that's shared is somehow a degenerate form of us or degenerate form of butterfly or whatever. So I don't mean to give a ladder kind of perspective here. But we always do include outgroups, so we can kind of see where the groups start from. Any other questions about this? All right, I just want to make sure, because we're not going to, in the next unit, behavioral genomics, you're going to see a little bit more of these phylogenies, and you should be comfortable with looking at them. You're not going to reconstruct them in this class, but when you read papers that have them, I want you to be able to sort of be comfortable with sort of parsing through what's going on here and what they're trying to say. And so in this case, I hope this story is sort of clear here. So basically, you can imagine the way this paper was written, they had different hypotheses for the relationship between, say, flowering plants and butterflies, or between butterflies and something else. And then they threw all these data into a computer, and it popped out this phylogeny, and this phylogeny that popped out rejected a bunch of other hypotheses that suggested that maybe butterflies that maybe you had, you could imagine a hypothesis where you need diverse butterflies before you get pollination services that produce diverse flowers. That's a totally reasonable hypothesis, but this phylogeny doesn't seem to support that. So, so that's how we can kind of use these phylogenies to test different phylogenetic hypotheses. All right, is that clear? We can get back to the octopus. All right, so we got our own, minor phylogenetic question here. Why do we have octopus eyes, or appear to have octopus eyes? So this is our thing that we want to test. So we're going to assume for the moment that this hypothesis is true. And we are going to build an experiment. So our experiment is, let's just cut open some eyeballs. Let's look at the structure of a bunch of cephalopod eyes, and let's look at the structure of a bunch of vertebrate eyes, and look inside them to see what they look like. So I'm introducing a new term here, experiment. What is an experiment? It is anything that helps you answer a what-if question. That's also my definition for a model. A model is anything that helps you answer a what-if question. And so we'll see why we have to answer that what if, where the if comes from here. But this is the experiment here is like, it's like that's the mystery. The what if here is like, what if we opened up the eyes of all these things and then compared them? What do you think we would get out? And that experiment is, well, let's just go and do it. And then once you've done it, you get an outcome. And that outcome can help you figure out if this is the right hypothesis. So once you have this experiment, your next step is to make a prediction. And that's the if-then statement. And the if of the prediction is the if in the what if of the experiment. So our prediction here is that if we do this experiment, if we assume this hypothesis is true, then when we cut open these eyeballs, we should see the same internal parts, roughly the same internal parts of the cephalopod eye and the vertebrate eye. And if we don't, then that experiment will have rejected this hypothesis, because this hypothesis necessarily would require this outcome. And so if this outcome isn't true, then this hypothesis must not be true. So an experiment's a what if, a prediction is an if then, and that's how they go together. So if I want to generalize that, then in all of these experiments, I'm going to have, a prediction, which will be if experiment, then the statement of the prediction, because the assumed hypothesis that we are testing. And so we test the if-then statement with the experiment, which helps us pose the what-if question. So we have a predicted outcome from the prediction, and we have an actual outcome from the experiment. And if those two don't line up, well, the experiment, we just did it. That's the real thing. So then we get rid of the prediction and thus get rid of the hypothesis. That's the method. Does that make sense? How all those fit together. Causal question, how and why. Hypothesis, a statement answering the how or why. Experiment, something that's a what if. And then prediction, an if then. And those all fit together in a certain way. Okay. All right, so if we're really clever, we can hopefully design one experiment that can test multiple hypotheses simultaneously. So we can say, one hypothesis, hypothesis A, will have this prediction under this experiment. Another hypothesis, hypothesis B, will have a totally different prediction under the same experiment. And so you can imagine that one of them might be common ancestor, the other one might be, not. The other one might be convergent evolution. And so under the common ancestor hypothesis, we get a prediction that the eyes are going to look the same. Under the convergent evolution, we get the prediction that the eyes might not look the same. So, so this is always, so like when we sit our PhD students down in their comprehensive exams, we ask them to give us all of their hypotheses. And then we ask them, all right, what would your prediction be? If that was true, what would be your prediction? Which was basically us saying, does your hypothesis mesh with the experiment you're proposing? And if it does, then under that experiment, what would you predict to be the outcome? And would that outcome be any different than all the other hypotheses? Because if the experiment you designed has all the same predictions across all the different hypotheses, you can't disentangle them. It's a bad experiment. So hopefully one experiment gives you different predictions for different hypotheses. And if you're really good and really lucky, hopefully you can design one experiment that tests a zillion hypotheses at once, where each hypothesis has a different prediction. So after you actually run that what if, you actually do the experiment, then it might knock out A, knock out B, not knock out Y, and knock out Z. And since you've knocked out A, B, and Z, and maybe everything else, then you'll then conclude that we can't reject Y. And so at the moment, because we don't have any other hypotheses, Y has the best support. All right, so any questions about that process? Yeah. Well, an example of an experiment that's tested multiple hypotheses. So, like I said, the cephalopod is one kind of example there, but you can imagine, you could say, for example, we're about to do a experiment about the honeybee brain, and so we're studying for so honeybees use both their eyes and their noses, their antennae, in order to seek out different food items. And so you can starve honeybees, and a bunch of things happen with the honeybees. And so the government is very interested in knowing how information processing systems in nature degrade when they're energy limited. And so we might imagine that a honeybee that's starved will become more discerning in its sensory systems because it's really important for it to find food. And that would be one hypothesis. But we might all alternatively suggest that actually a honeybee will become less discerning because it just needs to eat anything. And so it's not actually going to focus on a particular odor or visual cue. It's just going to go to anything as you make it hungry. So in one experiment, we can starve a honeybee. And so we can give it, a control case, an unstarved bee, and we can train it so that it knows that a particular odor is always rewarded or a particular visual cue is always rewarded. And then as we starve the bee, then we can see, does it focus more on that thing that it learned was rewarded, or does it stop paying attention to what it learned and just goes for everything? And so in that one experiment, we have two possible outcomes that either tell us if it's a variance seeker or a variance minimizer as they get hungry. Yeah. Well, so then the question is, what if there's other hypotheses? And that's really important when you do the experimental design, is to brainstorm with a team every possible answer that could possibly be, because ultimately you have to write this up and send it out to reviewers. And when you send it out to reviewers, you're going to have a reviewer that might say, hey, you guys tested hypotheses A, B, and C. You didn't think about D. And it seems to me like D would be consistent with your answers too. Why didn't you build an experiment that that is able to discriminate between D and A, B, and C? And your response sometimes is, I didn't think of it. So, and hopefully you don't do that. And so hopefully that's why you work with an experienced team and you sort of, beat things against a wall for a while, and hopefully you hit all of these hypotheses, because the last thing you want to know is somebody who just happens to come from a slightly different field and they look at it with fresh eyes and say, oh, your data could be explained also by this other hypothesis. Now, hopefully in that case, you've rejected enough to still make a contribution, but you'll just have to add a note to your paper that this also could be explained by this other hypothesis. And it's important in future work to design an experiment to disentangle those two. But we don't have to worry about these anymore because we already rejected those. So maybe it'll be easier to build an experiment to differentiate between the one we thought it was and the one the reviewer came up with. Any other questions? All right. So, so with that in mind, let's try this iClicker thing, huh? So hopefully all of you are able to join. And so you can either go to student.iclicker.com or open up your apps, and I will open up start the poll here. I'll start the class and then start the poll, multiple choice. All right, I'll give you a short period here and I'll capture this screen. All right, there you go. You should be able to see it. All right, 72 responses, 74, 80. All right, almost to full capacity here. I don't know if everybody's here, but wait for this to settle down. I think we got maybe our last response. All right, I'm gonna go ahead and hit stop. All right, so what we found there is that there was, I'll keep it running, there are a couple others coming in here. Most everybody got it right, where the answer, and remember, these are graded on completion, not correctness. But what I was looking for there is prediction. And we had, I think, a large majority of the class, so 87% you got prediction, but there's still a few, there's a sort of a side lobe here on experiment. So I can understand that. Remember, an experiment helps to answer a what if question, but it does not pose a then. The then has to come from assuming a hypothesis. And that's the prediction. So under this hypothesis, I would make this prediction under this experiment. So the if-then statements are predictions. They're definitely not hypotheses. And the experiments are what-if tools for answering what-ifs. All right, great. Okay, so back to the cephalopod. This is our experiment. We look at under this hypothesis, that would be our prediction. So let's go ahead and do that. We cut open a bunch of vertebrate eyes and cephalopod eyes. At first, they look kind of similar, but then we look a little more closely, and there's some craziness here. For one, the retina is in the wrong spot. So if you look here, the retina on the cephalopod is outside the nerve fibers. Here, the retina is kind of behind the nerve fibers. And then more importantly, the vertebrates have a blind spot. So in the cephalopod, because the nerve fibers are behind the retina, then it's possible to pull this nerve bundle out the eye without disturbing any of the retina. Whereas here, because the nerve fibers are on top of the retina, in order to get the nerve bundle out of the eye, you actually have to obscure some, or you have to create a hole in your retina so that you can't see on that hole. So there's a vertebrate blind spot, but there's no cephalopod blind spot. So, I mean, speaking of that, if you, how many people knew about your blind spot? Right, okay, so for those who haven't ever done it, unless you get queasy doing this sort of thing, if you want to see your blind spot, here's a simple way to do that. You can close, let's say, your right eye. You can put both thumbs in. This is so to look at your blind spot on your left eye. So I'm going to close my right eye, put both of my hands out front, and then I'm going to slowly move my left thumb away. And eventually I hit a point, a little hard with the projector here. If I focus on my right thumb while I'm moving my left thumb, then there's a point right here for me where my left thumb disappears. And that is my blind spot on my left eye. I can do the same for my right eye. And that is a feature of being a vertebrate, our crappy eyes. So cephalopods have a little bit of a better eye there, but we get around it. You know, it turns out it's a small enough area. For some people, it's larger, and some people, it's smaller. But it's a very different architecture. So that would suggest that we don't have a common eye. So that kind of rejects, or at least comes close to rejecting the idea that we had a common ancestor with this eye. All right, any questions about that? Okay. So that was one causal question we could ask about this whole thing. But then that sort of suggests other causal questions, like this blind spot thing, like, you know, how or why does a cephalopod eye not have a blind spot? And then a separate, how or why does a vertebrate eye have a blind spot? These could be asked independently. At this point, we're not comparing cephalopod to vertebrate necessarily. We're just asking, what is the process that generates a blind spot here? And what is the process that doesn't generate a blind spot there? So we can come up with hypotheses in each or one of these. So maybe one of these hypotheses is that the optic nerve is projected from the brain out. And because it has to shoot in, it has to sort of make a hole in the retina. Whereas here, the optic nerve is introduced initially, and then there's an invagination that kind of stretches things out so that you don't actually have to punch through this sidewall here. And then it turns out you can do an experiment where now, instead of looking at an eye now, you can look at an eye from early stages of development all the way up through the mature stage of development. And so I'm showing this here. This is sort of cartoon images for the vertebrate eye. And it turns out that you do get kind of this optic cup that forms, and then late in development, this optic bundle sort of shoots through the back of the cup, punches a hole, filters up there, and that's where you get the connections to your retina. And that's what creates the blind spot. But if you look at do a similar study with a cephalopod eye, then the sort of the nerves are already connected at the back from a pigment spot, and that pigment spot kind of gets deeper and deeper, and eventually fills out and gets a lens over it, and there's no blind spot there. So it's a totally different developmental process, and that generates a blind spot. So that's really, I think, interesting to me, that your development can totally change the function of your eye. Not apparently in this case too much, but it's like, even though we both need an eye, supposedly, the way an eye is built here generates a slightly different functional value than the way it's built here. And it's kind of like there's a bunch of features of hardware, like computers, cars, whatever, where you're like, why is there this funny little ridge? Like, you get your milk carton and there's like a ridge down or whatever. I'm like, what is that ridge doing for me? Like, occasionally that ridge, it's got a little plastic thing and it might scratch me or whatever. And that's really annoying. But that's how the milk carton is put together. These two sides end up being kind of fused together in this mold. And so it just happens to form a ridge. And I figure, it's still functional. So we're not going to worry about smoothing out that ridge. So, it's kind of a similar thing here. There's this sort of ridge, this blind spot that's created, and it doesn't cause a lot of problems, and it's more consistent with the vertebrate developmental process. And so it would be too hard to build this eye as a vertebrate because it just doesn't fit with all the other developmental stuff going on. All right, any questions about that? Remember, we're testing a developmental hypothesis now. So we were a phylogenetic hypothesis, now it's an ontogenetic hypothesis, a developmental hypothesis. Okay, all for the same animal. All right, so then kind of making this go a little quicker here. So then we can ask another causal question, another how or why question, and how do the pupils of the cephalopod eye constrict? How does that happen? So, what is the process that is happening, the mechanism that is making these eyes constrict? And there's a bunch of different things we could address. And one of the things we could address is how the two eyes relate to each other. And under one hypothesis, we can say the two pupils, there's a mechanism that couples the pupils together so that if you, I don't want to say if just yet here, that the constriction of one necessarily, that there's one process that is controlling the constriction of both eyes. So to test that, I could take a cephalopod in the lab and shine a bright light in one of its eyes and then see what happens to both eyes. And so I might see a constriction in one, a constriction in the other. I might see a constriction in only one. I might not see any constriction whatsoever. And it turns out for the cephalopod eye, they're not actually tied together. So if you do this experiment, I don't know if I've got that here. So if you do this experiment in the cephalopod, they can constrict the eyes independently from each other. So that is a mechanistic hypothesis there. It's how does it work? These two eyes mechanistically are not coupled together. Whereas kangaroo legs are coupled together. That's why you never see a kangaroo walking like this. Yeah. Say it again, the constriction. So I would say this is a constricted eye. So this is slightly constricted and this is totally open. So a cephalopod in the dark, their eyes look like this. A cephalopod in bright light, their eyes look like this. And a cephalopod in sort of intermediate light looks like this. Okay. All right, so that's a mechanistic hypothesis. The last thing when we look at these eyes, these beautiful eyes that we could ask is why do cephalopods need vision? Why do they have vision? What does vision give them? What utility do we have for vision? You could ask the same thing about vertebrates, humans, whatever. One hypothesis is it's all for sex. It's all for finding mates. So under that hypothesis, then I could build an experiment and this isn't necessarily a great experiment, but this is an example. I could put a bunch of cephalopods in a social setting together in light and a bunch of cephalopods in a social setting together in complete darkness. And then I could count how many mating events that I observe. And if there's a significant difference, then it seems like vision must be involved in mating. Alternatively, I could say, well, maybe it's not. Well, I don't want to say maybe it's not. Maybe they use vision to find prey. So I had to check myself there because both can be true, but I only test one at a time. And so I mean, I might end up testing both, but I'm not trying to say one is an alternative to the other or one is they're mutually exclusive. So in this case, I could do a similar experiment where I give them a bunch of foraging options, a bunch of little things that they eat in total darkness or in light. And then maybe that tells me that that if there's a difference that they are using their vision. But you might say maybe their prey is using vision. And if you make it really dark, then it's the prey that are having a hard time. It's not the cephalopod. So I could say, okay, well, a different experiment. What if I put a blindfold around a cephalopod? Well, how do I do a blindfold around a cephalopod? Well, like there are various animals. You can ablate their eyes. You can take their eyes out. You can paint over them. There's all sorts of things that people have done with animals to test to see if they're using their senses. And you can imagine doing an experiment like that. And in that experiment, that would only affect the the octopus and not its prey. So its prey would still have light, but the octopus would not. So an alternative experiment. So I'm trying to point out two things here. One, that a causal question can have two hypotheses that are both supported by the data. They don't always have to be alternatives. There can be multiple answers. So there can be multiple reasons why you have vision. The other thing I want to point out is that you can have multiple experiments for the same hypothesis. And every experiment might generate a new prediction. So the prediction under this experiment is this. The prediction under that experiment is that. So that's the other thing, is that like a hypothesis doesn't force one experiment. There could be many different experiments to test the same hypothesis. So there's a lot of multiplicity here. One causal question, multiple hypotheses, one hypothesis, multiple experiments, and thus multiple predictions. So any questions about that? Yeah. I don't know what you mean when it's not based on a morphological difference. Mm. Ah. And by related to each other, you mean how are you talking phylogenetically, or are you? Uh, yeah, so that can be that's again the clever experiments, and so a lot of times it depends on the particular case that you're looking at. So for these two particular animals, then you might cook up an experiment that is just peculiar enough to those two animals that allow you to sort of tease out that if there was a phylogenetic relationship, we would have this observable thing that would occur. Maybe it has to do with whether they can mate or not, or something like that. But it's hard for me to come up with a precise example. But I mean, that's a big part of the scientific method here, is coming up with good experiments. And those experiments, as you'll read, hopefully in some of these papers that you see in these AREs, the most clever experiments are the ones that take advantage of opportunities that are in the particular organisms that they're using. In fact, there is in integrative biology, we have a principle called Crow's principle, Krogh, which says that for every question, there is one or two or three, maybe at most, animals that are perfect for studying that question. And every other animal that you choose is just going to be a headache. And so a lot of times, if you're really interested in the question, if you're a question-driven researcher, the challenge is finding the right organism to be the model for that question. And because if if you just say like, oh, I want to study vision, I'm gonna, maybe ants aren't the best model for me to study vision, but cephalopods, maybe they are a good model to study vision. So, so it might be that if you're really finding a hard time testing your particular hypothesis with the particular organisms that you have available to you, maybe those are the wrong organisms to study this, unless you're only interested in those organisms, then you're just stuck with them. But if you're interested in the broader question and what does it mean to biology in general, then maybe you find another organism that's more tractable. Is that okay? Any other questions about this? All right. So we put all those things together, 4 different causal questions, all related to the eyes of the cephalopod and maybe how they relate to vertebrates. And then I've kind of ordered them here by the time scale at which the hypothesis is acting. So we've got like an evolutionary time scale. So this hypothesis acts over millions and millions of years. This hypothesis here, so this is the functional hypothesis. So this is, why the eyes popped up to begin with. Then millions of years, we've got the evolutionary hypothesis of how eyes evolved in relative to other eyes. Then the developmental hypothesis over the lifetime of an organism. How was that eye built? So, and then finally, the mechanistic question. At any instant of time, how does the eye work? And so I hope you can see that there's something qualitatively different about these 4 types of questions. And so this was sort of recognized as being kind of related to Aristotle's 4 causes. How many people have taken like a Psych 101, or sorry, a Philosophy 101, and you've heard about the 4 causes? Well, a couple, yeah. So this is like a standard sort of text about Aristotle, thought about causality, and he kind of grouped things into saying that you have the most kind of immediate causes, what things are built of, and then you have the efficient cause, what things built them, and then you have the formal cause, which is how did one thing change into another thing over sort of its history, and then finally the final cause, which is why does the thing exist? What does it do? And when behavioral ecologists looked at this, they said, it feels like, and one behavioral ecologist in particular, Nico Tinbergen, said it really feels like all of biology has questions, causal questions, that also fit into these 4 groups. So Tinbergen, who ended up winning a Nobel Prize for this work and others, that sort of founding the field of behavioral ecology, and one of the earliest, I would say, animal behavior as well. So he's Nobel laureate with Carl von Frisch, the honeybee guy, and Conrad Lorenz, the guy who had ducks that were imprinted on him, or not necessarily ducks, I don't know which fowl, some bird imprinted on him, then sort of said, well, I'll just rename Aristotle's causes. And I'll say, recognize that every biological causal question sits at one of these 4 levels of description. So it's, function, it's about whether it's, what is its adaptive value? What is its utility? What benefit does it provide? Phylogeny, where did it come from? How does it relate to other organisms? Ontogeny, how do you build it from scratch in each individual organism over its life history? And then finally, mechanism, how does it work at any instant of time? And so that's why I just went through these 4 cases for this cephalopod. And so to me, the way I view them is, it's kind of like function is like that final cause in Aristotle's term. It's driving everything, but it's pushed down. And because of the evolutionary baggage, there are a bunch of mechanisms that are off the table. Like I would like to move from point A to point B. It might be great if I had wings to get there. I'm just not going to develop wings. That's shot. I'm stuck with arms and legs. And that's going to be my mode of locomotion. There's no descendant of me that will have wings, And then ontogeny development, there are like some things like I'd love to have maybe a microchip embedded into my brain. And well, I don't know if that's actually ontogenetically possible. I don't know if it's possible for that to develop with my wetware. It just might not be possible. There's a bunch of mechanisms that, again, are off the table. And then that leaves you with a small set of mechanisms here that may not actually do a great job at giving you the function that you're looking for, but hopefully it's a good enough job. So to me, when I look at this, I think this is really a story of constraints, evolutionary and developmental constraints that ultimately shape the mechanisms that we have direct access to observe. And the beauty of that is actually diversity. And so we started this whole story talking about how the vertebrate eye has a blind spot and the cephalopod eye does not. And the reason why there's a blind spot in the vertebrate eye and not in the cephalopod eye is because they have different developmental pathways. And so that generated a diversity. If there were no evolutionary or developmental constraints, then you might imagine everything would look the same because there'd be selective pressure. And we can talk about drift and all sorts of other things that would perturb this idea, but there would be selective pressure for everything to be the best. And there might only be one best way to do things. And if things were totally fluid, then life would just evolve toward that. But life doesn't. Life gets stuck. And as it gets stuck in these ratcheting processes, then that's what generates the biodiversity that we have around us. And so I think the constraints are beautiful. The constraints give us the tapestry of life that we have around us. And that diversity makes ecosystems more robust, because they now don't have to be built on one design where if suddenly you shift the selective pressure out underneath you, everything dies. While every mechanism is not quite perfect, every mechanism is great at the job it was selected for and okay at some other job that it might be needed to do some other time in the future. So in the end, constraints, I think, are good for life. But when we ask questions, that's what we're doing. We're asking, how does it work? How is it constrained on a life history scale? How is it constrained on an evolutionary scale? And what is it actually providing to the organism? All right, so any questions about the four causes? So sometimes it's called Tinbergen's four. I've heard some people call it Tinbergen's compass. So like four cardinal directions. But every biological question fits into one of these. And we have to be careful not to confound them. So a lot of times people will be asking a mechanistic question and then suddenly they'll drift into making claims like, oh, the reason why the eye has, like if they say, well, how does the eye work? Well, they figure out how the eye works, then they immediately might jump and say, oh, well, that makes sense because that gives the eye the ability to see red better than any other eye or whatever. Like, but that's not the hypothesis you tested. You just tested how it sees red. You didn't test what the value of how it sees red is or anything like that. So you have to be very careful that you're only doing one hypothesis at a time, because you're going to probably have to do a different experiment at each level. And that different experiment is going to have different data sets. So rarely does an experiment here speak to a hypothesis there. Okay. Pretty clear? Okay, so yeah, questions? All right, so let's do one more of these. Oh, and actually, this is kind of not fair. Let me go back and then I'll tell you the right answer to this. But I meant to focus on this. This is my being out of practice. I will frequently in this class use the words proximate and ultimate. Proximate means closer to mechanism. So a proximate explanation is a mechanistic explanation. Ultimate means closer to function. So an ultimate explanation is going to be what value does it give? Roughly speaking, proximate are how questions, ultimate are why questions. So you can have proximate explanations, how does stuff work? Ultimate explanations, why is it around? Now, it's kind of a continuum because, phylogeny is sort of like an evolutionary mechanism, kind of. It's a little bit like a how question at a very long time scale, but it is definitely more ultimate than ontogeny and mechanism. So we can say, why do I have two arms and two legs? Well, I can have a phylogenetic explanation for that is a little more ultimate. It's not like a mechanism, as opposed to how do my two arms and two legs work? All right, so with that in mind, let's go to this other question here. And I'll kick off. Let me this guy. All right. And so there it is. So which of the four causes corresponds to the most proximate level of analysis? Is it A, function, B, ontogeny or development, C, mechanism, or D, phylogeny or evolution? So I kind of just said it, but let that go. Almost everybody in. And it's common to hear a behavioral ecologist when somebody's talking about a mechanism and someone starts talking about the adaptive value of that mechanism, even though so it's a lot of times people will say that a particular animal really likes the color red. Well, that's a mechanistic thing, and then someone might say, well, that's weird that they like the letter or like red because that seems like a crappy way for them to find food. Like, well, proximate ultimate, that's the whether that's a good way to find food is sort of immaterial. Like, I wasn't asking that question. I was asking, how do they find food? And they look for red. So, that's kind of what I mean by mixing. We want to be careful not to mix proximate and ultimate. All right, and so almost everybody got C. A couple of people did B, and one person did D. Function is definitely the most ultimate. So the rest of these are more proximate than function for sure. But the answer I was looking for was C for sure. All right, so then to close out today, this is a behavior course after all. So the question then is, why would an animal behaviorist care about the details of a cephalopod eye? What does that have anything to do with behavior? And this is meant to be a little bit of a prelude to Thursday, but we can then ask the question, well, what really is behavior? Does anybody have a good answer for how they would define behavior? Yeah. So personality, way you do so, it's sort of that's kind of a cognitive definition. Yeah. How you act or respond to stimuli. Okay. Anybody else? Yeah. So it's a predictable pattern of the way you act or react. I see. So if I hear a loud noise and I jump up and down, but the next time I hear a loud noise and if I do something else, then that's very different than if I hear a loud noise and always do the same thing over and over again. Yeah. Well, conditioning is a term, it has to do with learning, and we'll talk about learning. But learning, you could also say maybe behavior has to do with learning. Anybody else have a definition of what is behavior? How do you define behavior? What is the difference between behavior and physiology? Maybe a harder one there. Yeah. Okay, so it sounds like a hierarchy. You're sort of suggesting that behavior has to be built from physiology. So you should be able to have physiological explanations for behavior, but not necessarily the other way around, is how they might relate to each other. Anybody else have any other sort of, yeah, back there? All right, so that's kind of interesting that physiology sort of is how the organism maintains its internal state, but whereas behavior is how the organism responds to changes from the outside. It's an interesting distinction. Well, I don't have a great answer for what is behavior. There have been papers written about how if you ask experienced, distinguished behaviorists, none of them will agree on a definition for behavior. Marlene Zuk, who's sort of one of these distinguished behaviorists, she has studied a lot of sexual signaling, what the evolution of interesting sexual characteristics in animals, particularly conspicuous ones. Very well-known animal behaviorist, has a popular book that came out not that long ago, Dancing Cockatoos and the Dead Man's Test, the DMT, how behavior evolves and why it matters. So she's written a lot of, the academic literature, but this is a great book for the public about animal behavior. So, but in it, she talks about this dead man's test. And she was saying that drawing from the work of a psychologist, so how do the psychologists define behavior? And a common thing that comes up is this so-called dead man test. And it states that if a dead man can do it, it isn't behavior, but if he can't, then it is. So that's that's how they kind of cut it, is that is it behavior? Well, can a dead man do it or not? Sort of an interesting thought experiment. And then some people add to it. So a clinician added to the dead man's test here, anything a person says or does, we have to call a behavior. If it involves movement and has an impact on the environment, that's a behavior. If it's influenced by environmental events, that's a behavior. If it can be observed, described, and recorded, and then, of course, the dead man test, which apparently they sanitize sometimes and also call the teddy bear test, where it's the same kind of idea. Apparently, a teddy bear is an analog for a dead man here. And so that's, how they, the psychologists are defining behavior. But those definitions can also be kind of problematic. I mean, for one, like can be observed, described, recorded. Well, you can learn something and never act on that learning, right? So isn't learning feels like it should be part of behavior, doesn't it? I mean, talk about conditioning and things like that. Do we have to wait for an animal to act on its learning for us to be willing to call the learning behavior? It just doesn't feel, right. So, like, that's kind of the example here. That like, does it have to be immediately observable? Likewise, movement. It's fine, I think, to say if there's movement, it's behavior. But this require that all behaviors involve movement. Again, it doesn't really pass the learning test. You can learn not to react. You can not reacting is often the better behavior. Falling down and playing dead. That can also be a behavior, a very valuable behavior. So in this class, we're going to use kind of what Breed says, which is basically it's hard to define behavior. But if something includes at least one of these four things, movement, social interaction, cognition, or learning, we're going to call it behavior. But it doesn't have to include all of those. But if it includes one of those, then we'll call it behavior. Personally, when I think about the physiologies versus the behavior thing, to me, a physiological thing is something that doesn't really have a choice. So it is a metabolic pathway that once it gets turned on, it runs and runs and runs until it runs out of fuel. But to me, a behavior is something that is, going back to your point about physiology, it is physiologically possible for you to do more than one thing, and yet the animal does one of those things. To me, that one of those things could be do nothing. If it's possible to do something else, to me, I feel like that's a good enough description of behavior. But that's just my personal opinion. I'm fine with Breed's definition here too. So any questions about that definition? What we mean by behavior? All right, so questions in general? All right, so let's close then with I'll have one last attendance question right after this. Just a reminder, if you haven't done it yet, do those unit assignments in Unit 0. The first AREA and UCEA are due this Sunday, grace period until Monday. But again, there's a healthy drop policy in case you don't feel like doing it. And then otherwise, let's do one last poll here to close. And that's, and I'll see you Thursday. So let's do multiple choice. Do you think plants can behave? A, yes, B, no. No right answer, just sort of interested in seeing what you think. And after that, that's all I got for you. So feel free to take off. We'll see you Thursday. Okay. Hmm. Okay. So in that case, in the case of an animal choosing not to react, you would still say internally you should be able to sample, neuromodulators or whatever. And yeah, I think that that is a it sounds a much more precise way of what I feel like that yeah about the physiology. So yeah, I like that. That's good. Okay. Yes, yeah, from the left to right. Yeah, I can end it once that looks like everybody's right. No, no. Well, none of them are graded for correctness. They're just graded for whether you completed them. Yeah. Mhm. Uh-huh. Oh, I'm sorry. Okay. I don't have the slides before class. I think I just finished these, but I will, as I accelerate, I will post them to Canvas as soon as I have them. And then I'll go through the sales. I still am catching up with all the sales and but yeah, but yeah, to the best of my ability, the instant I have a draft, I will put it up on Canvas. Okay. Hmm. Uh-huh. Yeah. No, in that particular case, yeah, if you the we can come up with something, a paper based or whatever, or email. Yeah, that's fine. Uh-huh. Yep. I It's the class was recorded, so you can take a look. Is basically going over the course policies, but then also motivating, what we do in animal behavior. So there was kind of a behavioral example with some with a spotting behavior, and we kind of did some brainstorming. But otherwise, it just went over the course policies. So I would skim through the do the unit zero things, which basically go through the syllabus anyway, and then just skim through that video. Hmm. Yep. Hi. Let me just stop the recording here. So.